I'm going to be talking about how tumors, they disable the Psmac so that the immune cells cannot attack the cancer cells. So it's an immune evasion tactic that tumors like to use. So first, what the tumors are, so they're basically these cancer cells, but also why the tumors hate Psmac. And basically the Psmac is a formation which is the killing ring. So basically, it's anchors actin. So actin is a major a constituent or component of Psmac. So it transmits force. So the actual attack that immune cells use. That's why they need Psmac. So let's say a granzyme B perforin. So the outcome for the tumor, if the Psmac forms, which is why the tumor tries to disable the Psmac, is sustained immune contact, effective cytotoxicity, and immunological memory priming for the immune cells. Means at the end of the tumor. So tumors, they have multiple different ways and mechanisms of destabilizing the Psmac. One of the first ones is high acetyl CoA conditions boost cholesterol synthesis. Therefore, tumors, they load excessive cholesterol and store excessive cholesterol into the lysosomes, or cholesterol is even incorporated into the multivesicular bodies, or the MVBs. The way tumors do that is with high acetyl CoA, which is from excessive sugar with protein. As a result of the cholesterol loading, the tumor intentionally does that so that the lysosomal membranes are too stiff, making them less able to fuse with the MVBs with the exosomes, because the MVBs have the exosomes loaded into them, and if the MVB fuses to the lysosome, then the tumors cannot get the exosomes. But the tumors need the exosomes in order to destabilize the Psmac. So that's why the tumors load the lysosomes up with cholesterol so that the lysosomes cannot degrade the exosomes. So as a result of cholesterol accumulation inside the lysosome, or the MVB, this accumulation promotes exosome formation instead of lysosomal degradation. These exosomes can carry enzymes or mRNAs that reprogram immune cells. So if tumors receive those exosomes, the tumors will be able to modulate the immune synapses remotely, and those are basically, the synapse are the anchoring or the bridging points where the immune cells have to stand and dock on and not slip off. But the tumors, if they get the exosomes, that they'll be able to cause the immune cells to slip off, because the synapse will become slippery, and it's not supposed to be. And that's what tumors can do with the exosomes. So tumors can disrupt the Psmac's peripheral adhesion ring in the immunological synapse. And tumors do that by using the exosomes in several different ways. So the exosomes, if the tumors receive the exosomes, in the exosomes are loaded with these soluble things called ICAM1 or PDL1. And if the tumor gets the exosomes, they use, let's say, the PDL1 will bind to the T cell receptor. What that ends up doing is that it weakens the adhesion ring, or the Psmac itself, because the Psmac is basically like a sort of death ring against the tumor. Okay, tumors with the exosomes, they can also prevent the perforin granzyme B release, or it's basically the attack that immune cells use against cancer cells. But the tumors can stop that by using miRNAs that are delivered, or that were loaded in the exosomes. Also, another way that tumors like to destabilize the actin ring at the Psmac. So the Psmac is made of actin itself, is by it occurs if the tumor gets the exosome. In the exosome, our signaling molecules are loaded into the exosomes. So miRNAs, also RHOGT pace. So these destabilize the Psmac. As a result of this, the T cells cannot sustain contact on the cytoskeletal actin surface. So the exosomes, they can even carry or be loaded with metabolic modulators that are similar or alike to acetyl CoA precursors, which can be really problematic because acetyl CoA normally forms from high sugar, high protein uh, diets, or high sugar, high protein consumed together. But it seems like tumors, they can make their own acetyl CoA without even the human consuming high sugar, high protein. So as a result, T cell contacts the tumor, but the Psmac can't form. The granules don't polarize, and these are needed for perforin granzyme B killing. So that fails, and then eventually, so the tumors effectively trained immune cells to not kill the tumor. Okay, so a good way to defend against this is to target the PDL1 that's loaded into the exosome, or even possibly stop it from loading in the first place, because the PDL1 destabilizes the Psmac, because the PDL1 binds to the ligands or the receptors on the T cells, which has to be prevented. Therefore, the Psmac has a better chance of forming. A good food here to stop PDL1 is red cabbage, has anthocyanins, sulforaphane, a vitamin C, polyphenols, fiber, indole 3 carbinol. So all those have a very good effects, especially vitamin C and sulforaphane, but it's beyond the scope of this video. Just going to focus on PDL1. Okay, so PDL1 is often driven by NFKB. So that's CD27, CD70 axis. So that axis can also be broken, such as with a pineapple. Pineapple is a protease. It cleaves the adhesion molecules that keeps the CD27, CD70 on. By cleaving those molecules, the connection is broken, therefore NFKB is reduced. So PDL1 is also driven by STAT3. Another good food in combination with red cabbage is a papaya. Papaya can also lower STAT3. Papaya also raises STAT5 to help the immune system, but that's a different rabbit hole for a different video. So another really critical way that red cabbage helps suppress PDL1 is by modulating it at the epigenetic level. Because sulforaphane in red cabbage is known as a DNA methylation modulator. So basically that means that PDL1 expression is actually reduced at the chromatin level or at the DNA. Because if anything that expresses PDL1, such as the gene for PDL1, PDL1 won't work without its gene that it needs. But sulforaphane at the chromatin level will reduce that gene that drives PDL1. Therefore, no PDL1 to destabilize Psmac. Also, PDL1 is driven by ROS, but anthocyanins in red cabbage lowers reactive oxygen species. Therefore, no PDL1 upregulation. That's it for the video. Like, comment, share, subscribe. Also, leave your own knowledge and mechanisms down in the comments to fill in any gaps, and I'll see you in the next video.